Have you ever thought about how good are we in curing disease? Well, actually, we are really bad at it. How bad? Well, to date, we know about 70,000 diseases out there, but actually we can cure less than 1%. But don't worry, we are constantly trying. But we're not just trying like this, that we're all happy and joyful. We are trying more something like this, that it's a really long and frustrating process. In the past 70 years, we try every year thousands of different drugs, but annually we are successful in only 25 drugs that are approved by the FDA. Because it takes a lot of time and a lot of money to develop a drug. How much time? About 20 years. And how much money? About $2 billion. Well, so what is the $2 billion question? That what hasn't changed in the drug development in the past 70 years? Well, we know the technology changed. We know that we were able to land in the moon in the past 70 years. We know that we developed sophisticated electric cars. Our knowledge also changed significantly. We characterize the human genome. We can do quantum computing. So what haven't changed? The answer is the drug development process itself. At the end of the day, every drug that is being tested on humans is being first tested on animals. And although there are many, many similarities, we have similar organs, we have similar physiology, we can even share a lot of the same DNA with the animals, for example. We share about 95% of the DNA with rodents. But at the end of the day, between 60 to 90% of the drugs that actually pass animal trials fail in human clinical trials. Because at the end of the day, most of the humans are not rodents. So, in order to make it a successful process, what do we need? Well, we need human models, but we don't have many volunteers, right? So, $2 billion question is, can we create a human model that is not a human being? The answer is yes. Yes, we can, and the way we do it is by using this novel concept known as organs on a chip. Organs on a chip is a way that we use actually tissue engineering to mimic the organ functionality in a Lego-like device. For example, the idea is that we have a kidney. The kidney, at the end of the day, is a filter. So what we do, we take human kidney cells and we create a filter out of this. And then we put this inside this plastic USB stick-like device, what you actually see here behind me. And we call it a kidney on a chip. I will give you another example, the heart. The heart roughly is a pump. So imagine we take human heart cells and we create something that contracts like a pump. And we put this inside this Lego-like chip, and then we will call it a heart on a chip. And this is basically what you can see behind me. You can see these heart cells that contract like a real heart. And we can do it for many different organs. But at the end of the day, we know that an organ doesn't have only one functionality. An organ can be very complex. For example, the lung is not just gas exchanger. We know, for example, it has an immune response. We know it controls the temperature. We know it can help to create sound. It has a complex physiology. It has complex anatomy. And how we can mimic all these different functionalities and characteristics inside this Lego chip. So the first organ that was developed by Dan Hu and Don Imberg at the Visa Institute, and was basically this lung on a chip. These chips can be very sophisticated. You can have multiple cells inside. As you can see here, you have the lung cells, the endothelial cells separated by a membrane. These chips can be elastic, so we can actually mimic the movement of the lung. We can bring air on one side. We can bring blood at the other side. And we can even do more sophisticated things, such as to bring immune cells and bacteria and to see the immune response of the body inside these chips. But when this project started about 10 years ago, there were individual chips. And when I saw this new technology, I said to myself, wow, we definitely need to try to integrate them to make it as a human, small human on a chip. So I joined to this team basically to lead this integration of multi-chips by creating this mini human on a chip. And you can imagine that this human on a chip is like a Lego. You have the heart on a chip and the brain on a chip and the gut on a chip, and you can start linking them together to create this mini me on a chip. And I would like to give you an example how such a system enables us to look at the systematic effect, how the human body responds to different conditions. And I will give you an example with the organ that is rarely used. In this case, I would like to give you an example on the brain. So what you see here is an amazing video that was taken by the group of Ed Boyden at MIT. And you can see here an image of a rodent's brain. All the green lines that you see are actually neurons inside the brain. You can see the complexity. You can see the amazing structure. You can see the amount of cells that you have inside. But what I would like that you will keep in mind is that what you saw here is half of a mouse brain, half of what you see here at the back. And for most of us, the human brain looks something like this. A lot bigger, a lot more complex, and the human brain at the end of the day has a unique structure, unique functionality, and also very unique with its disease. For example, we have Alzheimer, Parkinson, and many other diseases that do not occur in rodents. We ask many rodents if they have Alzheimer, and none of them remember the answer. So in order to mimic this complex organ, what we do is we look at the anatomy of the brain. And here you can see we have blood vessels, we have the brain cells, the neurons, and we try to use the chips to mimic this complex system. And as you can see by the image, there is a huge difference between these plastic chips and this anatomical system. So how can I claim that we can use such chips to mimic such a complex system? So actually what we do, we can take these chips and we can link them, like I showed you previously with the Lego, and we can actually see the different interactions that you have inside the brain, the blood vessels and the neurons and different things. And we can actually use the system to identify and to check, for example, how toxicants affect the brain, how disease and bacteria affect the brain, and how drugs affect the brain. But now that I show you how we can break the brain by using the chips, let me show you how actually we can breaking bad using the chips. And what we can actually do is to see the effect of methamphetamine, drugs on the brain, similar to the TV show Breaking Bad. So here you can see an image of cells. The cells that you can see at the bottom that look like a carpet are basically the blood vessels, and the cells that you see on the right are the neurons, the cells of the brain. What we can do is to bring methamphetamine, crystal meth, to the system, and you can see how suddenly the blood vessels are erupted. They look really bad, actually, and you can see suddenly how some junk and toxicants go into the neurons. So the take-home message of this is that, yes, we can use the system to learn about drugs, but more importantly, that you shouldn't take drugs because it is messing up your brain. Okay, so and now that we talked about some ideas how you can tackle the challenge of drug development, I would like that you will keep in mind that the problems in drug development do not stop here. It's true that there are significant differences between animals and human beings, but also there are significant differences among humans themselves. So just to give you an idea about the statistic is that at a given time, 75% of the people take medications that are not optimal to their condition. Moreover, ideally, we would like to have a platform that will enable us to have a personalized medicine, that each one of us will be able to get the medicine that is best for his or her condition. And to tackle this, what we can do, and we do today in our labs, is to take a sample from someone's skin and to transform these skin cells to be any type of cells of the human body. And by this, we can create a specific tissue which contains your own DNA and to create your own personalized medicine. And imagine we integrate this with the previous technology. Like this, we can create actually a mini-me on a chip that will integrate our different organs with the different chips with our own DNA and our own specific physiology. We can then test and identify drugs for different diseases, right? We can study corona, how it is affecting our own body. We can study neurodegenerative disease. We can study cancer. We can study bowel disease, and many, many more. But I would like to show you just a small example of what we can actually do in the lab regarding to neurodegenerative disease. So what we do is we take a biopsy from a Parkinson patient. We transform these skin cells to be brain cells, neurons, and we can actually measure their electrical activity. This electrical activity that you see here is similar to the electrical activity that is actually measured with EEG. So we can learn in a dish in our lab things that are similar to where you go to the clinic and you will do EEG. But remember, keep in mind that we don't have one cell or one sample. We can do multiple. We can do many of them. And for each one of them, we can start to do drug screening. So now we can take your own mini brain and to give drug A. If it doesn't work, we give drug B. If it doesn't work, we give drug C, etc. And you can see the Lego again. We can create drug screening for your own disease. And by this, you will have your own personalized medicine. This will actually minimize the time you need to wait for curing your disease. And at the end of the day, by this, we hope to create the future of medicine, that you will go to the doctor, you will take a biopsy of your skin, you will create your mini-me on a chip, we will do drug screening, and at the end of the day, you will have your own personalized medicine. And we hope that with this, we can take this poor statistic from 1% to much, much higher, and to change the way we do medicine today. Thank you.